All right, good morning, everybody. This week is our last week of lectures, and we're going to cover two pathways, okay? And I'll try to emphasize the important things that you need to know. Similar to the other pathways we've done, really it's a general overview in terms of what the pathway's doing, what molecules it needs, what molecules it produces. So if we're talking about gluconeogenesis, we're making new glucose. So we're generating that, right? And that's going to be an anabolic pathway that we'll look at. We'll look at what substrates can be used, and what's really important is that you appreciate that this is going to supply glucose for your brain. Okay, your brain is always going to be needing glucose to generate energy. It can't use fats for energy, so it does grab whatever glucose gets to it. It's got glucose transporters that glucose just gets in automatically so that we can always be supplying your brain with energy. We'll see where that lactate's coming from. Dr. DeMello's mentioned that already, and we'll talk a bit about, we'll review again how it's considered a waste product and how it can get used in gluconeogenesis. And going back to the beginning of the second half, talking a bit about thermodynamics and how that drives some of these pathways. Okay, and as always, that regulation is going to be really important. So we'll look at a couple scenarios, things like diabetes, and how it can lead to the production of glucose in gluconeogenesis. All right, so again, this simplified map will be on the final exam. So when you get into the exam, feel free to jot down all the little points that you want to remember, and then you can use it as you're going through the questions for the final exam. All right, so let's just start off with a little bit of review, a little bit of integration. So I'll put a question up on Mentimeter. So in terms of low oxygen, what effect is that going to have on the body and sort of the biochemical pathways? Okay, so let's just do a quick review. So maybe take 30 seconds or a minute with your friends. Oh, I don't have the Menti code up. But log on to Mentimeter and answer the first question that's on Mentimeter. All right, what effect does low oxygen have on metabolism? We've got a lot of people saying lactate can be catabolized to generate energy. That's our top pick. There's some people that are saying there will be no proton gradient for ATP synthase. Okay. Now a lot of people are picking LDL oxidation will be inhibited or gluconeogenesis will be stimulated. All right, so let's just take a look. If you picked B, you are correct. Lactate's going to be made, but it's not going to be catabolized. So that's kind of the key point, okay? So remember your ETC needs O2, right? It's going to be the final electron acceptor to produce a water molecule that pumps H+ for ATP synthase. So if we don't have any oxygen, then we're not going to, hang on a second. We're not going to be able to pump H+, right? And so we're going to have no ATP made. Okay. Lactate is going to get made, right? So anaerobic glycolysis will make lactate. But the whole point is so that NAD+ is going to get made so that glycolysis can continue and we can make those two ATP molecules. All right, so let's just switch over to the next question. Okay. What about under starvation conditions? Okay, so when you're going through these pathways, think about what happens in sort of the normal state, right? When you're breathing properly, you're getting lots of oxygen, we're making lots of ATP. What happens if we're under the starvation state and we need energy? Which of the following pathways are going to be stimulated? Okay, what about under starvation conditions? All right, so if we're under starvation conditions, we need to make ATP. thinking about all of your pathways, which pathways are going to be on and which ones are going to be off. So let's take a look at Mentimeter. Come on. All right, so we've got a couple pathways. We've got a couple of pathways, couple of enzymes. So we've got glycogen synthesis, right? So if we want to be making energy, glycogen synthesis is going to be off. We've got acetyl-CoA carboxylase. What does acetyl-CoA carboxylase do? Yeah, in the back. So acetyl-CoA carboxylase is involved in fat metabolism of some sort. But you're saying breakdown? Okay, so when fats get broken down to acetyl-CoA, that's in beta oxidation. Okay, and so we didn't learn the names of all of the enzymes in beta oxidation, but it's actually not beta oxidation. Somebody had their hand up here? Acetyl-CoA carboxylase over here, yeah? Turns acetyl-CoA into malonyl-CoA. So that's actually the start of our fat synthesis pathway. Okay, so glycogen synthesis is going to be off. Acetyl-CoA carboxylase is actually going to be off as well. How are we going to be making our energy? We're going to be making it in glycolysis by breaking down any glucose that's present. So phosphofructokinase is going to be on. Creatine kinase, we did talk a bit about how it can help make ATP, but it's a very limited store of phosphates, right? Creatine was that molecule that's in your muscle cells. It's a limited source. There's only going to be about a couple seconds of ATP. So if you're starving, you're really going to need to get all that energy from your fats, so from glycogen, fats, and eventually proteins too. Okay, so our glycolysis is going to be on. Our PDC will be stimulated, beta oxidation with the import of our fats to make acetyl-CoA, and then that's going to be used in the citric acid cycle. All right, last question. What about glucose homeostasis? Go back to Mentimeter. Why is glucose homeostasis going to be important? Yeah? I'll let you think about that for a second. So one other thing I'll just put on here too, just based on a point. So glycolysis is going to be on in other cells, like in all cells to make energy, right? But today we're going to talk about your liver cells. Your liver cells are going to be special. They're going to be making glucose. So they're going to generate glucose in GNG. And so that's what we're going to look at today. Right? So you have to think about like different cells, right? Whenever the glucose comes to your brain, it's going to be broken down and used in glycolysis. Right? And so when it comes to why is glucose homeostasis important, right? We're preventing hypoglycemia as well as hyperglycemia, really generating that energy, okay? And that's why our body is going to use the liver. It's going to help supply glucose for things like your red blood cells and your neurons, right? These are all going to be dependent on glucose. They can't use fats. So we are going to have to generate glucose, right? So we're going to avoid hypo versus hyperglycemia. And we're going to supply our cells with glucose, right? Whether it's red blood cells or your neurons that are primarily dependent on glucose. Okay, and so that's just a little bit of review to kind of set up why we're talking about gluconeogenesis, why we have this pathway that's going to be in our liver cells that are going to help make glucose and supply glucose for the rest of your body. So when we look at gluconeogenesis, okay, it's almost the reverse of glycolysis, but it is going to be slightly different. Okay, so we're going to be starting with a variety of different molecules, and so things like acetyl-CoA can't actually be used, but we can start with our pyruvate. So acetyl-CoA was kind of our central molecule that can get used to generate fats, but there are other molecules like glycerol. Glycerol can get used in gluconeogenesis, as well as pyruvate and lactate and some other amino acids. So we're going to talk a little bit about how those can actually get used. So we started talking a bit, our discussion today in terms of this idea of limited oxygen, right? So if the electron transport chain isn't running, it doesn't have that final electron acceptor, then anaerobic metabolism is going to occur. So everything that's happening in your mitochondria, here's just an electron micrograph of our mitochondria, all those processes in the inner membrane and the matrix aren't going to be able to occur. So instead, what's going to happen is glycolysis, which is out in your cytoplasm, your cytosol outside, is going to be the only way of generating ATP. So we had this additional enzyme that Dr. DeMello talked a bit about, lactate dehydrogenase, which sometimes is considered like the 11th step after glycolysis. So glycolysis had 10 steps. Lactate dehydrogenase is this 11th step that's going to run under low O2 conditions, or if you have problems with your mitochondria. And what that's going to do is it's going to take the NADH that was made in glycolysis, and it's going to convert it back to NAD+. So we have this redox reaction where NADH is going to get oxidized, and our pyruvate is going to get reduced to form lactate, and that's going to be considered our sort of waste product. And now that NAD+ is going to go back into glycolysis at step 6 and allow it to run again so that we can generate maybe 2 more ATPs from each of our simple monosaccharides. Okay, so really important mechanism to allow just a little bit of ATP to be made, but unfortunately now we have this sort of waste product, lactate. Lactate levels are going to rise, and if you see elevated levels of lactate, say in your bloodstream, that could indicate that anaerobic metabolism is taking place. That lactate can then get used in what's known as the Cori cycle, and Dr. DeMello did show a version of this slide, a slightly different one, but you can go back and start to make these connections between the material when you're studying. So if we're talking about red blood cells that don't have any mitochondria, or say if you're exercising and you're not breathing properly, and your muscle cells aren't getting enough oxygen, then that's when anaerobic metabolism is going to kick in. We're going to convert our pyruvate to our lactate. That NAD+ is also going to get made. That can go back into glycolysis, right? So that we can continue to make those 2 ATPs. And then that lactate can actually get used by the liver in an anabolic pathway that we call gluconeogenesis, where it's going to first get converted back to pyruvate. So we actually see the reverse reaction taking place. So the lactate will get picked up by the liver. Lactate dehydrogenase is going to convert it back to pyruvate. And then pyruvate is going to enter into the gluconeogenesis through a series of steps in order to generate glucose. Okay. Your liver can then export that glucose, and then it can get used by other cells. Go back to those muscle cells, red blood cells, or even those neurons that we were talking about. So I think it was, I just wrote it down, lecture 18, Dr. DeMello also has a version of this slide, so go back and take a look and make sure that you can understand how all these pathways are kind of coming together. Right now we have this substrate. If we have elevated levels of lactate, right, under anaerobic conditions, now we have this other molecule that the liver can use in order to generate glucose in gluconeogenesis. So this is going to take place in your liver. Your kidney also does about 10% of all the gluconeogenesis. So whatever glucose that's being synthesized in the body, the majority is coming from your liver. So it's definitely a really important organ in your body that's going to help make glucose. It's kind of like the provider for the rest of the body. And really important because your brain can't use fats. So we're going to be solely relying on glucose to generate ATP. Okay. We've got substrates like lactate. There's also that 3-carbon glycerol that we talked a bit about. So remember when your tags get broken down, the fatty acid chains can't get used to make glucose, but that glycerol backbone can. Okay, so there's 3 carbons in there, so you're going to require 2 of those to make our 6-carbon glucose molecule. And then there's also going to be different amino acids that can also get turned into glucose. Okay, so these are known as glucogenic amino acids. So they all have these carbons in them that we can use to create that 6-carbon molecule. And it's all going to be in this anabolic pathway, so we're going to require lots of energy. So we're going to take a look at where that energy is going to come from. So if we compare our 2 pathways, okay, this slide is just showing the 10 steps of glycolysis in blue. So if we were to start at the top, again, you don't need to memorize all 10 steps, right? But we have some important steps that are sort of highlighted in yellow. These are our 3 regulated steps, right? So we start with glucose, we go through stage 1 of glycolysis to break our molecule into 2 3-carbon molecules, glyceraldehyde and 3-phosphate and dihydroxyacetone phosphate. We have 2 of those that are going to go through the second half of glycolysis, right? And this is where our NADHs and our ATPs were going to get made, right? So that's going to run twice. When we were looking at carbohydrate metabolism, we looked at the thermodynamics. So we saw that it was a very favorable pathway. So whenever that glucose comes in, we've got these irreversible reactions that's going to help drive the pathway and drive the catabolism of glucose. And we're going to get lots of free energy released. If we want to go in the reverse direction, you would have to put in 84 kilojoules of moles of energy, right? So it'd be a lot of energy that we'd have to put in. And so instead, what we're going to do is we're going to try to bypass these irreversible steps. So instead of trying to reverse them, you would need a huge amount of product, which is now your substrate, to make them go in the opposite direction thermodynamically. Instead, what we're going to use is we're going to use different enzymes. So there's going to be 4 enzymes. These are just highlighted in pink. These are going to replace those irreversible enzymes in glycolysis. So in glycolysis, steps number 1, 3, and 10, these are definitely steps you need to know and how they are regulated. When we want to make glucose, instead we are going to use different enzymes that are going to bypass these 3 steps. So pyruvate kinase was step number 10 in glycolysis. Instead, there's going to be a 2-step reaction to convert pyruvate to phosphoenolpyruvate. Then we just go through our reversible steps in glycolysis. So we're just going in the opposite direction. So your products are now going to be your substrates going through the pathway. That second half is going to run 2 times, and then we're going to join 2 molecules together to form fructose 1,6-bisphosphate, and then we have 2 new enzymes, fructose 1,6-bisphosphatase and glucose 6-phosphatase, that are going to remove the phosphates, eventually making our glucose molecule. Okay, and just showing here, here's our glycerol, just starting sort of halfway, coming through, and again, you would need 2 of those, or 2 pyruvates, or even 2 lactates in order for gluconeogenesis to run. All right, so we'll just look at the reaction mechanisms of these new enzymes. All right, so the first step in gluconeogenesis, this was the reverse of step 10 in glycolysis. We're going to start with our pyruvate, right, which could be coming from lactate. And what we're going to do is we're going to add on a CO2 group. So pyruvate carboxylase, looking at the name of the enzyme, so the carboxylase tells you it's an enzyme. It's using pyruvate as its substrate, and we're adding a carboxyl group onto it. Okay, so bicarbonate is going to be the molecule that's going to donate your CO2 group, and we're going to require an ATP molecule. So one of the first steps where we're requiring energy for our anabolic pathway. That's going to make oxaloacetate, which we've seen quite a few times now. Remember, it's important for the citric acid cycle, and now it's also a molecule that can get used to make glucose in gluconeogenesis. So that's the first step. Then we're going to have step number 2, where we take oxaloacetate and we're going to decarboxylate it. So we've seen this a couple times in our other pathways as well. And when that spontaneous CO2 group leaves, then we're going to actually join our phosphate onto oxaloacetate to form phosphoenolpyruvate. Okay, and the enzyme that does that is PEP carboxy kinase. So it's going to be a kinase. It's transferring a phosphate over in order to form phosphoenolpyruvate. Okay. Where else have we seen this mechanism? So if you do more work into metabolism or start studying different pathways, you might see similar mechanisms happening. So can anybody recall this mechanism where we used bicarbonate, we stuck a CO2 group on it, and then we use that as the driving force to make something else? Where else have we seen this? Yeah? What's that? Making malonyl. So that's, so when we're talking about our fat synthesis, right? So for fat synthesis, we saw that we had acetyl-CoA, where we added a CO2 group to make malonyl-CoA, right? And then that went into fatty acid synthesis. Fatty acid synthase uses that same mechanism, that decarboxylation that we're seeing down here, where we were decarboxylating a molecule, and that was helping to drive the synthesis of our fatty acid chain. Okay, so similar mechanisms that you might see throughout different pathways as you go on in your studies. Okay, and again, right, it's really not about memorizing every single enzyme, but if you can appreciate similarities that you see between things, if you can start to use this knowledge to apply it to other problems or other scenarios, that's really what will help you understand what you're learning about, okay, and what you're doing with that knowledge. All right, so that was our first irreversible step. So the last step in glycolysis is actually going to be a 2-step reaction in gluconeogenesis. We're going to go all the way through the reverse steps of glycolysis. So we're going to have 2 of those PEPs are going to get made. They're going to go through the reverse sequence of enzymes in glycolysis. Remember, those are reversible based on the concentrations of molecules. So if you have lots of these products, they're now going to get used as substrates to go in the opposite direction. In glycolysis, we had a cleavage reaction where we broke our 6-carbon molecule into 2 3-carbon molecules. In gluconeogenesis, we're going to join them. We're going to join 2 3-carbon molecules in order to form fructose 1,6 bisphosphate. And then an enzyme called fructose 1,6 bisphosphatase. So phosphatases are always going to use a water molecule to remove a phosphate from carbon number 1. All right, so here's our, remember, go back and remember how we are labeling our carbons, right? 1, 2, 3, 4, 5, 6. The phosphatase is going to use a water molecule, and it's going to hydrolyze off the phosphate from carbon number 1 in order to form fructose 6 phosphate. Then again, we're going to go through the isomerization reaction that we saw in glycolysis to form glucose 6 phosphate. And then glucose 6 phosphatase is going to come along, and it's going to hydrolyze off the phosphate on carbon number 6. So there's our C6 in order to form our free glucose. And now that glucose can leave the liver. Okay, so it's going to leave your liver for other cells to use. Where else have we seen this enzyme? Glucose 6 phosphatase. Where else? Yeah. The opposite. Okay, so whenever we are in the liver and we are breaking down our glycogen, right, we're also going to see this enzyme. Remember, we were starting with glycogen. We were using PI in glycogenolysis or glycogen breakdown to form glucose 1 phosphate. Then we had our phosphoglucomutase that was mutating, that was changing its structure to form glucose 6 phosphate. And then your phosphatase, again, is going to use a water molecule in order to hydrolyze off that phosphate to form glucose. And now again, your liver is now sharing that glucose with the rest of the body. Right? So muscle cells keep the glucose and use it for energy. Liver cells are going to release the glucose for other cells to use as energy. All right? So one enzyme that's going to help with 2 different processes where the liver can make glucose, either from glycogen breakdown or from gluconeogenesis. Right, and your liver's, it's a fairly big organ, but there's not that much glycogen in it, right? It will be able to supply some glucose, but then when the liver eventually runs out of that glycogen, that's when gluconeogenesis can kick in and can help make glucose for the rest of the body. All right, so if we were to start with pyruvate, okay, there's going to be 2 pyruvates that are going to be required, right? Each of the pyruvates have 3 carbons on them. So we have 2 3-carbon molecules to make our 6-carbon glucose. We're going to require a huge amount of ATP. We're going to require 4 ATPs, 2 GTPs, and 2 NADHs. Okay, so these are the high-energy molecules that are going to help drive this anabolic process, going from 2 smaller molecules to a bigger molecule. We're also going to require 6 water molecules in order to make that glucose. Okay, and there's going to be 11 enzymes that are going to make a single glucose molecule, but it's going to be overall favorable when you have lots of energy present. All right, so I'm going to put up a Mentimeter question. What I want you to do is I want you to take a look at the pathways and see if you can figure out where the ATPs are coming from. Okay, where are these 4 ATPs going to come from? All right, so we've got a couple correct answers and a couple, okay, ATP synthase, yeah, they probably did originally come from ATP synthase at some point, but I mean within the pathway, when you're starting from pyruvate in order to generate glucose, where are those ATPs coming from? Okay, so Yes, we're going to require them at pyruvate carboxylase. Okay, so that was one of the reactions we just looked at. So that was one of our new reactions in gluconeogenesis. So it's going to require an ATP, and that reaction is going to happen twice. All right, so just backing up. Here was our first ATP that's going to be made, and this is going to happen 2 times, right? So you're going to have 2 pyruvates coming in, and we're going to require 2 ATPs at pyruvate kinase. And then on Mentimeter, somebody has also mentioned 3 phosphoglycerates, so let's just go back and take a look. We'll just back up. Again, you don't need to know the names of the enzymes, but if we look at our pathway, right, remember when you are talking about glycolysis, there were steps that made ATP. So anywhere where you make an ATP, if we're going in the opposite direction, you're going to require an ATP. Okay, and so those steps are going to happen 2 times. So we're going to have 2 ATPs required at that step, and then those 2 that we just saw at pyruvate carboxylase. Okay, so because stage 2 is happening twice in reverse, we're going to require 4 ATP molecules. Same thing, we saw that PEP carboxykinase was requiring GTP, so there's going to be 2 GTPs that are also going to get required as we go through those reactions twice. And then our other high energy molecule was NADH. So in glycolysis, we make NADH. Gluconeogenesis, we require NADH. All right, so these are going to be the steps where those energy molecules are going to be required. So any step that's reversed from glycolysis and something's made, it's going to be a substrate where it's required in gluconeogenesis. Okay, and then you could also go through and you could track the waters being used for hydrolysis as well as reverse steps in gluconeogenesis. Okay, and then that bicarbonate also comes from a water molecule as well. All right, so just overall requirements, you need lots of energy for this anabolic pathway. Okay, so we did that. Oh yeah, I guess I should have put that up. Okay, so just go back and take a look and make sure you understand where they come from, right? So any reverse steps, reverse, or the new steps, they are going to be substrates. Whereas in glycolysis, they were products. Right? We made 2 ATPs in glycolysis, 2 NADHs in glycolysis, and 2 waters. All right, so when we're talking about glycerol, so this is a little bit slightly different because it comes in halfway through our pathway. So remember, your fatty acids can't be used to generate glucose because your PDC is essentially irreversible. So that acetyl-CoA can't get converted back to pyruvate by the PDC. But the glycerol backbone that we released from our triacylglyceride molecules, so your tags, remember they have a glycerol backbone plus 3 fatty acids. We can use 2 of those glycerols in order to generate glucose in the liver, right? So it also has 3 carbons, so 2 of them will combine to form that 6-carbon glucose, and they're all going to enter in as dihydroxyacetone phosphate. Okay? It's also going to be an anabolic pathway, but we don't need as much energy, so it's a little bit more efficient in terms of making that glucose. You're only going to need 2 ATPs to add phosphates on, and then we're also going to make 2 NADHs. We'll still require those 2 water molecules to remove the phosphates at the end of gluconeogenesis. So looking at this pathway, again, you don't need to know all of the steps, but I just wanted to show them so you have an appreciation of what's happening, right? We start with our glycerol molecule. We are going to have a kinase that's going to transfer a phosphate on from one of our first ATPs to form glycerol phosphate. That's going to happen 2 times, so that's where our second ATP is going to get required. And then there's going to be a redox reaction where a dehydrogenase is going to produce those NADHs as well as our dihydroxyacetone phosphate. One of those will get isomerized to form glyceraldehyde 3-phosphate, and then we start to go through the reverse of the last couple steps of first steps of glycolysis, removing the phosphates again to make that free glucose. Okay, and then that glucose will get released into the bloodstream. All right, so when you're going back and studying, take a look at your pathways. Do that compare and contrast, right? Glycolysis is going to be our catabolic pathway where we are making ATP and NADH if you're under aerobic conditions. Gluconeogenesis is going to require ATP, GTP, and maybe NADH. So we'll just put a little star next to the NADHs, right? Because it really depends on whether you're under anaerobic conditions or what starting molecule you're starting with in gluconeogenesis. Glycolysis is going to produce pyruvate or even lactate under anaerobic conditions, whereas gluconeogenesis is going to make glucose. That's going to be our final product that's being made. And then where are these pathways going to be active? So glycolysis is going to be active in all cells, including liver, right? Your liver can still break down glucose to generate energy because it itself needs its own energy. So glycolysis can be on in the liver. Then once we have lots of energy, and if we need to supply the rest of the body with glucose, that's when gluconeogenesis is going to kick in your liver as well as in your kidneys, about 10%. Glycolysis is going to be generating energy for those cells, whereas gluconeogenesis is supplying blood glucose for the rest of the body. And then when are these going to be active, right? Glycolysis is always going to be on when glucose is available and we need to make energy, whereas gluconeogenesis is going to get turned on whenever you're exercising or maybe if you're fasting and you don't have a lot of blood glucose. Okay, and we'll look at the regulation of that after the break. All right, so we're really just talking about sort of this reverse pathway where we're generating glucose and then maybe that glucose is going to go out into the bloodstream for other cells to use. All right, one thing I do want to just talk a little bit about before we take a short break is this idea of amino acid catabolism. Okay, so over here on the right, I just have some little cartoons representing our 20 amino acids. And your proteins can also get broken down to generate energy, right? Or if you are eating a high protein diet, those amino acids are going to come into your body. Your body's first going to use them to make all the proteins and the enzymes that your cells need to function. But if you have excess amino acids, they can also get used for energy. So they can come in at various points to form the various metabolites and eventually generate ATP in oxidative phosphorylation. We don't have time to study the urea cycle, but it's another example of a cyclic pathway where you have multiple reactions that repeat. And the whole point of that is to get rid of that nitrogen component. So your amino groups on your amino acids are going to get excreted through the urea cycle. But the carbons themselves, they can come in and get used for energy. They can also get used to make glucose. So some of your amino acids can actually get used to produce, say, pyruvate, and then that can enter into gluconeogenesis as well. Okay? So I just want to kind of tie it all together. Again, you don't necessarily need to know the structures of your amino acids, but I do want to pop them up and see if anybody can kind of recall, maybe see that integration in terms of our 20 amino acids. Which ones do you think could be used to produce glucose? Okay, and how does what's really going to be their journey through the various pathways? All right, we've got lots of votes coming in for alanine, which is great because alanine definitely is one of the amino acids that can be used to generate glucose. Okay, and alanine is actually on this slide. So here's our alanine, okay, number 1. And some of the other amino acids that people were clicking on were aspartate and glutamate. So these are 2 others. Okay, and again, it's not about memorizing these at this point, although hopefully you can recognize your side chains. That's going to be really important for your assignment, as well as any future courses where you're looking at protein structures. Okay, but these molecules can actually come into our pathways. And again, you don't need to recognize or identify what molecules these are on the right, but I just wanted to show you how similar they are to the amino acids, right? So you can kind of appreciate how do these amino acids get into the various pathways. So when it comes to alanine, which molecule do you think it gets used to make? 1, 2, or 3? Hold up your hands. 1, 2, or 3. Which one do you think alanine is going to get used to be made? So we got some 3s over here, right? So if we look at its structure, we see similar structures to this one. Okay, and so this is actually our pyruvate And when we remove that amino group, so remember when we're catabolizing or modifying our amino acids, so this would be a deaminase, we're removing that amino group and we're going to replace it with our C double bond O down here in order to form pyruvate. All right, so that's one way it can come into the pathway. We can then convert the pyruvate back to glucose. All right, what about our aspartate? Okay, so similar mechanisms, right? We can see that aspartate has the 4 carbons, 1, 2, 3, 4. Over here, our second molecule as well also has 4 carbons. Okay, so that's another way we can get an amino acid into the pathway, right? And this is actually oxaloacetate or OAA. Again, with the deamination, and then glutamate can come in to form this molecule. This is alpha-ketoglutarate. Right, and so these are all in the citric acid cycle. So they can help produce energy if we need to generate energy from our amino acids, or we can use them to go through gluconeogenesis. There's the names. All right, so if you were just trying to trace the path, right, alanine comes in to produce pyruvate, and then I'm just going green. We're going to make pyruvate. It's going to go to oxaloacetate through to gluconeogenesis to form glucose. We had our aspartate that was producing oxaloacetate. Again, that oxaloacetate can then get used to make glucose. And then your glutamate was coming in to form alpha-ketoglutarate, which is going to go through your citric acid cycle to form oxaloacetate and then form glucose. Here's some chatter going on back here. Is there anything I can help with? Are we curious about what's going on back here? Yeah, question. Great question. Can glutamine and asparagine be used in the cycle? Yes, definitely. Right? Remember, asparagine and glutamine were very similar in their structures when we were talking about those non-essential amino acids, how we can make one from the other, for sure. Yeah, you just need one extra reaction where you convert that asparagine to aspartate, then goes to oxaloacetate, or your glutamine goes to glutamate, and then that goes through alpha-ketoglutarate all the way through. Okay. How are we doing up here? Are we okay? Yeah? All right, we're going to take a quick break, and then we will come back and talk about regulation of these pathways and glucose homeostasis. All right, let's come on back and talk a little bit about regulation. Right, so this pathway gluconeogenesis is specific to the liver and kidney, but I'm going to stop saying kidney. It's just think about it in your liver, okay? It's a pathway that's on in your liver, but glycolysis can also be on in the liver, right? Your liver cells are cells. They need energy. They need to grow. They need to divide to carry out whatever function they want to carry out. One of the functions could be gluconeogenesis, right? So the liver needs to kind of decide which one's going to be on at any given time, okay? So glycolysis could be on, gluconeogenesis could be on, glycogen synthesis could be on, glycogen breakdown could be on, and so this is when we need to start talking a little bit about regulation, okay? Because they can both be on in a liver cell, but they're not going to be on at the same time, okay? You would have this futile cycle of making glucose, breaking it down, making glucose, breaking it down, right? So we have to have a way of regulating these steps. So when it comes to the things that you're reviewing for your final exam, glycolysis is on the left here, so this is step number 10, okay? So step number 10 in glycolysis, as you were going through and reviewing and looking to see how it can be regulated. Remember, glycolysis is going to make energy. So if we need energy, if we're in the low energy state, AMP can stimulate step number 10. We also had a molecule called fructose 1,6-bisphosphate. This was coming from earlier on in the pathway. So this was an example of feed-forward regulation to stimulate the last step. So we had a molecule from earlier on in the pathway telling the last step, hey, you need to hurry up. We've got a lot of things backing up in this pathway. You need to speed up this pathway. We had ATP that was inhibiting the last step, right? If you already have lots of energy, we don't need to break down that glucose, and maybe we could store that glucose as glycogen. Similarly, if you have lots of acetyl-CoA from fats, so if your fats are getting broken down, we can make lots of energy from our fats. If you have lots of proteins or amino acids, those can also come into the pathway like we just saw to generate ATP, and that can also inhibit glycolysis so that maybe we can save the glucose as glycogen if you have lots of glucose present. If we need to make glucose for the rest of the body, okay, we have our anabolic pathway. So going in this direction, we have our anabolic pathway that needs energy to run. So ATP needs GTP, we saw already as well. So when we look at our enzymes and we look at how they are regulated, if you don't have a lot of energy, so if your ATP levels are rising, we can inhibit these two steps, right? So low energy inhibits anabolic pathways, high energy stimulates it. So we have substrates that are required. We can also use acetyl-CoA. Break that down to make ATP, or that molecule itself can also allosterically regulate pyruvate carboxylase. Okay, so this is a form of allosteric regulation. All right, so anabolic pathway needs energy. If you don't have energy, we're going to inhibit that two-step reaction in gluconeogenesis. So that was one level of regulation. Now looking at one of our other irreversible steps in glycolysis, this was step number 3, where in glycolysis we took fructose 6-phosphate and phosphofructokinase 1 transfers a phosphate onto carbon number 1 to form fructose 1,6-bisphosphate. Same trends. If you need energy, AMP can allosterically regulate phosphofructokinase 1. If you have lots of energy, you can turn off glycolysis. Okay. There was this other molecule called fructose 2,6-bisphosphate. That was made by PFK2, and it can also stimulate PFK1 and glycolysis. If we look at the opposite enzyme, so in gluconeogenesis, we are taking off the phosphate, so the phosphatase is going to remove the phosphate from carbon number 1 to form fructose 6-phosphate. The opposite is true. Low energy inhibits, and so does this fructose 2,6-bisphosphate molecule. Okay, so compare and contrast. Look at one pathway, look at what stimulates it, right? Low energy stimulates glycolysis, inhibits gluconeogenesis, or this molecule, fructose 2,6-bisphosphate, can stimulate glycolysis and inhibit gluconeogenesis. All right, so now what we need to do is we need to kind of finish the story about this molecule, fructose 2,6-bisphosphate. All right, so let's take a look at it. So it's an allosteric regulatory molecule. So here's its structure over here on the right. Again, it's not about memorizing what this molecule looks like, but it's being able to number your carbons, being able to describe where the functional groups are, right, where are your phosphates. if they're on carbon number 2 or carbon number 6. So being able to label and number your carbohydrate structures are going to be really important. Okay. And when we make it, it's going to stimulate PFK1 in glycolysis or inhibit our gluconeogenesis enzyme. When Dr. DeMello introduced this molecule, we told you that it was made by an enzyme called PFK2, right? Phosphofructokinase 2, because it was adding that phosphate onto carbon number 2. This enzyme is actually really cool. Okay, we haven't looked at it or talked a bit about it, but if we look at it, it actually has 2 domains. Okay, so it's a single polypeptide chain. Actually, it's got 3 domains. So here's sort of a representation of the sequence. Okay, so it's got 470 amino acids in its primary sequence. When those that polypeptide chain folds, you can see lots of alpha helices. There's a couple beta strands forming little beta sheets in there. And when those secondary structures come together to form our tertiary structures, we have different domains. So we have a kinase domain and we have a phosphatase domain. Right? So this is a tandem enzyme with 2 very different roles. One role is a kinase. So the kinase is going to add the phosphate. And then there is a phosphatase domain that removes the phosphate. So this enzyme is going to be important for controlling the formation of 2,6-bisphosphate as well as the breakdown or hydrolysis of 2,6-bisphosphate. Right? So it's a single polypeptide chain that folds to form 2 very different active sites, and it's all going to get regulated by phosphorylation. So there's this other stretch in its sequence. So at the N-terminus of the protein, there's a stretch called the regulatory domain, and this is where a phosphate can actually get added on. So we can covalently modify the enzyme, and that's going to change its activity. Okay, so there's this idea of reciprocal regulation that's going to be really important in order to phosphorylate the enzyme, and that's going to tell it whether or not the kinase domain is on or the phosphatase domain is on. Okay, and when the kinase domain's on, we're making fructose 2,6-bisphosphate. And when you have fructose 2,6-bisphosphate, you're going to stimulate PFK1 and glycolysis. If the phosphatase domain takes over, removes that phosphate, then it's no longer stimulating glycolysis and inhibiting FBP1. All right, so let's just walk through this diagram. Okay, so if we start on the left here with fructose 6-phosphate, here's our tandem enzyme that is going to transfer the phosphate onto carbon number 2 to form fructose 2,6-bisphosphate. Okay. So your tandem enzyme, when it is in the dephosphorylated state, so here's our serine 32 in that regulatory domain, there's no phosphate on it, so your kinase is going to get active. So that's what the little star is. Here's our active kinase domain, transferring a phosphate from ATP onto fructose 6-phosphate to form fructose 2,6-bisphosphate. Okay, now we have that regulatory molecule, and it can stimulate glycolysis through PFK1. All right, what happens if we want to change the activity of this enzyme? Right. We can phosphorylate it. So protein kinase A is going to come along. It's going to transfer a phosphate from ATP onto your regulatory domain. So serine 32 is going to get phosphorylated. We get an overall conformational change. So now your kinase domain is going to be off. Okay, so your kinase domain's off. And now your phosphatase domain's going to be active. It's going to take a water molecule, hydrolyze off that phosphate, and now we have our fructose 6-phosphate that could then get converted to glucose 6-phosphate and then even glucose in the remaining steps of gluconeogenesis. Right? And then that can go into the blood. Right? So we're no longer inhibiting gluconeogenesis, and instead we are allowing it to make glucose for the rest of the body. How does that phosphorylation occur? It all comes back to your hormones, right? So glucagon, remember that was our low blood glucose hormone. So under low blood glucose, glucagon signaling is going to signal through our GPCRs. That's going to activate protein kinase A, which is going to phosphorylate and turn off the kinase domain. And so now glycolysis is off and gluconeogenesis can be on. There's one other enzyme too that's going to get turned off. So if you remember pyruvate kinase, so that was step 10. So step 10 in glycolysis, it's also going to get phosphorylated and turned off by glucagon signaling. Okay, so two ways of slowing down glycolysis in the liver cells. We're going to slow down step number 10 in glycolysis. We're also going to remove this allosteric regulator, fructose 2,6-bisphosphate, remove that so we're no longer stimulating glycolysis, and now gluconeogenesis can take over. Right, so your GNG is going to be active, and your liver cells can release that glucose for the rest of the body. If our GPCR signaling pathways are going to be phosphorylating and inactivating these enzymes, then your insulin signaling pathway is going to do the opposite. So remember, in our insulin signaling pathway, we had phosphatases that were coming along and dephosphorylating enzymes. The tandem enzyme as well can get dephosphorylated, and now our phosphatase domain is going to be off, and your kinase domain can take over, again, making that regulatory molecule to stimulate glycolysis. All right, so two hormones, our low blood glucose hormone, our high blood glucose hormone, both have the ability of reciprocally regulating this tandem enzyme in order to either turn on or off your kinase domain or your phosphatase domain. Okay, so definitely go through and make sure you understand your signaling pathways, what enzymes are turned on or off by phosphorylation. If you're making a list, your glycogen metabolism enzymes, your fat metabolism enzymes, definitely go through and compare and contrast them, because this is one other way of regulating whether glycolysis is on or gluconeogenesis is on, all through this allosteric regulator, fructose 2,6-bisphosphate. All right, so in the liver, we have reciprocal control to make sure that both pathways are not on the same time. So we're either breaking down glucose to generate energy or synthesizing glucose in gluconeogenesis. We see hormonal regulation through that signaling pathway where we are either phosphorylating or dephosphorylating different enzymes. So make sure you know all of those target enzymes and whether they're on or off when they're phosphorylated. We have this tandem enzyme. This guy is kind of a unique enzyme in that it has two different roles. So phosphorylation will actually control the breakdown of fructose 2,6-bisphosphate, versus dephosphorylation allows for the production of fructose 2,6-bisphosphate. And then we also see energy charge, which is a little bit more intuitive if you don't have a lot of energy. So decrease in energy is going to stimulate glycolysis through step number 3 and 10. And it's also going to slow down gluconeogenesis through its enzymes, right, our two that overcome the irreversible steps, as well as fructose 1,6-bisphosphatase. Whereas if you have lots of energy, we can inhibit glycolysis in the liver cells and stimulate gluconeogenesis. All right, so I'm going to put up another Mentimeter question. Hopefully you have it open. Sorry, I keep forgetting to, I forgot to put the code on these ones. Oops, not that one, we did that. All right, so thinking about a liver cell, how could you rank the pathways in terms of sort of priority? What's a liver cell going to be doing? What's the first thing it's going to try and do versus what's kind of the last thing that it's going to try and do? All right, so we've got glycogen synthesis and glycogen breakdown or glycogenolysis have kind of been switching. But let's go with this order that's here, okay, in terms of our order. So we'll just go 1, 2, 3, and 4. Okay, so let's think about this in terms of after a meal. Okay, so we're gonna eat some carbohydrates. Those carbohydrates, we're talking about the liver. Okay, so we're gonna talk specifically about the liver. Remember, the liver's got membrane transporters called GLU2. That GLU2 is going to allow our glucose to come in. So we're going to have glucose import, right? And our default is going to make ATP. Right? Because a liver cell, like any cell, needs lots of energy to divide and carry out all of its function. All right, now that we've made all that ATP, right, that ATP can now inhibit glycolysis. Remember, we can take our ATP, we can transfer our phosphates onto UDP to make UTP plus ADP, right? And then that's going to help with glycogen synthase. All right, and remember, for glycogen synthase to be on, we also have our insulin signaling. And then that's going to dephosphorylate glycogen synthase in order for it to be active and use our glucose. So remember, we were going from glucose 6-phosphate, we had the isomerization, to glucose 1. Right? Then we activated it to UDP glucose, and then that's what glycogen synthase was using as our substrate for glycogen synthesis. All right, so lots of regulation, kind of walking through what's going on in those cells. All right, so we filled up all of our glycogen. We're no longer storing glucose. It might get released into the body, used by all the other cells, and then we might end up switching to our catabolic state. So if we're in our catabolic state now, right, where we want to maybe supply that glucose for other cells, so our glucagon, GPCR signaling can take over, right? We get our protein kinase A that's going to activate glycogen phosphorylase. Via phosphorylation. We also get our tandem enzyme. I'll put that up under gluconeogenesis in a second. Just take that out. Okay, and then we're going to produce our glucose 1-phosphate, and then that can go to glucose 6, and then glucose for the bloodstream. Or our gluconeogenesis, last thing, if we have lots of ATP, right, we can turn off glycolysis. So we have pyruvate kinase that's going to get phosphorylated. We've got our tandem enzyme that's going to get phosphorylated. And our fructose 2,6 bisphosphate is going to go to fructose 6 to glucose 6 to glucose. Okay, so we're going to be making lots of glucose using some of that ATP that might be generated perhaps maybe from fat cells as well. Okay, so lots of different ways that the liver cell can help supply glucose to the rest of the body, so helping maintain that glucose homeostasis for the rest of the body, right? And then that glucose leaves the liver cell, and then it gets used going back to number 1, glycolysis in all other cells, right? The glucose is going to come in, and now we can use it to generate energy in those cells. All right, so we're going to just talk a little bit about diabetes, because the discovery of insulin, I'm sure you've heard about it in your biology classes already. Big discovery at the University of Toronto. There's a memorial, I guess, with lots of historical features over in the Mars building. So if you're kind of, we're over here, Con Hall's over here. If you go down College Street, kind of by Queen's Park subway station, you can check this out if you're ever over on that side of campus. But I do kind of want to talk a little bit about insulin and these hormone signaling pathways, because it is really relevant for biochemistry. So talking a bit about homeostasis, again, important for diabetes in humans, Feline diabetes can also occur, so some people might have to give their cats insulin as well. Unfortunately, Garfield got killed off. I don't know why they did that, but it was kind of funny. He's just a cartoon, but maybe they were trying to bring awareness to feline diabetes. So I'm kind of curious, what do you already know about type 1 versus type 2 diabetes? So when it comes to these two conditions, maybe chat with your partner, take 2 minutes, and see if you can recall anything about these two conditions and sort of their differences on Mentimeter. All right, so we've got lots of ideas coming in. So a lot of people are aware that you can be born or develop with type 1 diabetes versus type 2 kind of develops over time. Or it can be sort of acquired, right? We have a lack of insulin production with regards to type 1 versus insulin resistance in type 2, so insulin resistance. Sometimes you might hear insulin insensitivity. So that just means cells are not responding. So that insulin signaling pathway we talked about, where insulin binds to the receptor and then ends up auto or transphosphorylating the receptor in order to activate enzymes inside of the cell. Because you have way too much insulin present, those hormones, the receptors are no longer responding to those hormones. Okay, so in both cases, we don't have any insulin signaling. So again, for type 1, there's also going to be no insulin signaling. But it's really because of that lack of insulin present versus the receptors themselves not responding. Okay, so this is our receptors not responding or not signaling versus no signal to begin with. Okay, really that difference between the two. So relate it back to the biochemistry that you're learning, right? Insulin itself is also, I don't have it. This weekend I was going to bring it. I found a toonie. I don't know if anyone has seen these toonies. So I found a toonie from 2021. They put out an insulin commemorative toonie, and it actually has the structure, the protein structure of insulin on it, and it's in the color blue, which I thought was kind of cool. Blue is the color for ribbons to support diabetes. So I was going to bring it to show you, but I totally forgot. But it was just kind of neat to see it. I know nobody carries cash anymore, so it's not like you have your change. But when maybe if you're out and you actually have toonies, take a look and see if you can find it. I'll try to remember on Thursday. I totally forgot to bring it. All right, so relate it back to the proteins and what you're learning in biochemistry. Okay, and so we've talked a lot about how insulin's important for the recruitment of those glucose transporters. Dr. DeMello talked a bit about carbohydrate structure, and glucose itself is very reactive. Okay, so here's the structure of glucose, okay, down here. And if you have hyperglycemia, so hyperglycemia means high blood glucose. If the glucose isn't getting used to make energy or stored as glycogen, that's where hyperglycemia can arise. And the reason why this condition is not great is because the glucose can react with lots of other proteins or lipids or even nucleic acids. Okay, and so here's one example of where glucose actually reacts with hemoglobin. So here's the N-terminal valine on your beta chains of hemoglobin. So again, hemoglobin was a tetramer, 2 alphas, 2 betas. Each of the betas have free N-termini. And that aldehyde group, so remember this CHO, this is your C double bond O, H, very reactive. Glucose can cyclize, so make sure you go back when you're studying for the final exam. You know how glucose can be a cyclic structure. So this is our linear form. But it can form your alpha structure, your alpha anomer, and your beta anomers. Okay, but when it's in the linear form, Instead of that aldehyde reacting with the hydroxyls, so remember for cyclization, we had these hydroxyls that were attacking the aldehyde. Instead, what they can do is they can react with lone pairs of electrons on our amino groups to form these shift bases or the more stable Amadori product, where we just move that double bond. Okay, so this is a spontaneous reaction that can happen. This is what we call glycation. Okay, so you don't need an enzyme. It just happens spontaneously. Lots of glucose floating around. That glucose comes into proximity of different proteins or lipids, and this reaction occurs, and now you've modified the structure, right? So now we have hemoglobin with this sugar molecule stuck on it, and this is what we call hemoglobin A1C. Okay, so when I was showing you my blood work, that was one of the tests that they were doing, checking to see how much hemoglobin A1C was present, and if there's elevated levels, this tells you that your blood glucose was high sort of over the past couple of months. Okay, so your red blood cells get turned over every 4 months. And so if the hemoglobin that's present in those red blood cells are getting modified, then this might be one way of noticing whether somebody has been hyperglycemic over a long span of time. Okay. There's other molecules that can get glycated, so these advanced glycation end products, just another way that glucose can react with other molecules and might actually modify and affect their function. Okay, so hemoglobin can still carry oxygen. It's not affected by this glycation, but other molecules can get modified and can lead to some of the negative symptoms that we see due to hyperglycemia. So this is where synthetic insulin can come into play for type 1, right? So this, I think someone on Mentimeter had talked a bit about treatment, right? Treatment for type 1. We can help administer insulin, right? So as blood glucose levels rise, if somebody has type 1 diabetes, they're going to take some insulin, maybe before a meal. That's going to help recruit your glucose transporters, so those GLUT4s for your muscle cells, right? In order to use the glucose in glycolysis, generate some energy, and then that energy can get used in order to synthesize maybe your fats, your proteins, or glycogen as well with excess glucose. It's also going to turn off your alpha cells. So alpha cells, those were the ones that were secreting glucagon, right? And we're also going to turn off our catabolic pathways. So our glycogen breakdown, our beta oxidation, as well as our gluconeogenesis, because we don't need to make any more glucose. The problem, though, is if you take too much synthetic insulin. Okay, so say somebody with type 1 diabetes takes their insulin and then forgets to eat the meal. Right? So there's no actual blood glucose coming in to get used. That insulin is going to be the stronger hormone. Okay, so in terms of regulating those beta-adrenergic receptors and the glucagon receptors, we talked a little bit about which one was stronger when we were talking about signaling. Insulin leads to the degradation of your receptors, and it also affects the secretion of glucagon, so we can no longer signal for that drop in blood glucose. Okay, and so if you take too much insulin, then that's when someone's blood glucose might drop, right? We're still telling the cells to take that glucose in and use it in glycolysis, and we no longer have access to any of the glucose in glycogen breakdown or gluconeogenesis. Okay, this is all due to that hormone signaling by insulin. Okay, so that's one reason why hypoglycemia could arise in someone with type 2 diabetes. Can you think of maybe two more? When you think about maybe some of these pathways, some of the biochemistry, see if you can come up with two more ideas of why hypoglycemia might arise. All right, we've got some answers coming in. So took insulin without eating food. So we just talked about that scenario, right? If there's no carbohydrates coming in, then you're going to see hypoglycemia. Exercise definitely is going to drain that glucose, use it all up for energy. Your glycogen's also going to get broken down, so that can lead to hypoglycemia if we're no longer able to supply it. I think somebody else mentioned where we skipped meals in general, right? If you're not consuming any carbohydrates in the first place, then that's going to inhibit it as well, not provide any glucose. Where was it? There was one. Injecting too much insulin for sure. Oh, here we go. Mutation with gluconeogenesis. So it's always not on, but always off. Okay, so we've got sort of skipped meals, right? Too much insulin. No carbohydrates. We were talking about exercise. Okay. Someone mentioned gluconeogenesis, so there could be problems with gluconeogenesis, right? So if gluconeogenesis isn't on, so if it's off, then your liver's not going to be able to supply any glucose to the rest of the body. There could also be problems with glycogen. With your glycogen stores. So if you can't break down that glycogen and share the glucose with the rest of the body, then that could be another issue that arises. Okay, and so both of these we could kind of put together with liver function. So some people might have different blood tests done to check their liver function, and if your liver's not functioning properly, then it's not able to supply that blood glucose for, say, your brain. Okay, so there could be issues with liver function. I think somebody also meant glucose urea. Let me just go back. So someone had mentioned, or glycosuria. So this would be high blood glucose. But when we're talking about that, that could be your kidney function too. Okay, so your kidneys, there could be issues with kidney function in terms of excreting glucose. So that could definitely be something. So renal issues, kidney issues, either for insulin clearance maybe, or glucose levels too. Any issues with your kidneys could also lead to hypoglycemia. All right, so when it comes to somebody with type 1 diabetes, really balancing how much insulin's coming in, that anabolic hormone, with the carbohydrate intake to ensure those can get used, as well as exercise and that requirement for energy. When we have that insulin injection, that's going to help with glucose uptake. Remember, go back and review those glucose transporters, right? Some of them are insulin sensitive. Some of them don't need insulin signaling in order to uptake glucose into different cells, but that's also going to turn off your glucagon signaling. And these are going to both play a role in hyperglycemia. So compare when hyperglycemia arises, okay, in terms of signaling and how they can turn on and off the different pathways, such as gluconeogenesis, that could also worsen your hyperglycemia. Okay? As you're studying and going through these pathways, think about what's happening in your body after you have a meal, or if you haven't eaten in a while. If your blood glucose drops, what's going to happen if you have insulin present versus the other hormones and the various pathways that are taking place. Okay? And then on Thursday, we're going to look at ketogenesis, so another pathway that can help with energy production in the body. Keep up with your review. Just a reminder, your final exam is going to be a combo of multiple choice questions as well